Hello, welcome guys. We are meeting after a very long time. Hi, this is Dr.JIBRAN from Medical College Hospital, Kolkata, and I'm absolutely sorry that I was late. I had some exams before, because of that I couldn't upload videos, but now I can upload some videos. I would wish to highlight few points for this videos. As you have seen in my previous videos, all of my notes were handwritten and put down, but for these videos, subsequent videos are all digitalized, made in PowerPoints, and I hope that I'll make it more easy for you people to grasp. So without wasting any more time, let us move ahead. So let us see what are today's topics. Okay, so topics for today is that we will read about selected examples of necrosis and injury, including ischemic cell injury mechanism, ischemic reperfusion injury, chemical injury. We will read about the role of mitochondria in cell death, apoptosis in details, necroptosis, as well as pyroptosis. Let us look at this very simple schematic taken from Robbins. Now we see that what is the role of mitochondria in cell injury and death? Let us see. What is the role? First, we see that whenever there is a reduction in oxygen supply, that is hypoxia, toxins or radiations, it leads to what is called as mitochondrial damage or dysfunction, that leads to reduced ATP generation and increased production of reactive oxygen species, leading to multiple cellular abnormalities, ultimately culminating in necrosis. I repeat, whenever there is hypoxia, toxins and radiation, that leads to mitochondrial damage or dysfunction, leading to a reduced ATP generation with increased production of reactive oxygen species, ultimately leading to necrosis. On the other hand, let us look at the other mechanism that of cell death that is there, or where mitochondria is playing a role. So whenever there is a reduced survival signal, or there may be irreversible DNA or protein damage, it leads to increase in the amount of pro-apoptotic proteins and decrease in the amount of anti-apoptotic proteins, leading to leakage of mitochondrial proteins, mainly cytochrome C, which will lead to activation of caspases, which ultimately leads to cell death by apoptosis. Let me repeat. Whenever there is reduced survival signals or DNA or protein damage, it leads to an increased pro-apoptotic proteins as well as reduced anti-apoptotic proteins, leading to leakage of mitochondrial proteins, namely cytochrome C, which will lead to activation of caspases, leading to apoptosis. Coming to mitochondrial damage, as we've already discussed, any kind of hypoxia, toxins, radiation can lead to mitochondrial damage and dysfunction, leading to decreased ATP production and increased ROS production, leading to what is called as necrosis. Also, necrosis there is damage to the cell membrane as well as damage to the membranes of the organelles, for example lysosomal damage, ultimately leading to cell swelling as well as enzymatic digestion. Look, the other mechanism where mitochondria is playing a role in cell death is whenever there is reduced survival signals, as we have seen before, DNA or protein damage, it leads to an increase in the amount of pro-apoptotic proteins and decrease in the amount of anti-apoptotic proteins, ultimately leading to the release of cytochrome C, which stimulates the caspases and then ultimately leading to apoptosis. Going to the next slide. Irreversible cell injury, as we have already seen, has two main components. Number one, there has to be a damage to the mitochondria leading to loss of mitochondrial function. And number two, there has to be membrane function abnormality. I repeat, irreversible cell injury is having two major components. There has to be loss of mitochondrial function and there has to be a membrane function abnormality. Now, ischemia, as we all know, is the most common cause of cell injury. I repeat, ischemia is the most common cause of cell injury. How is it different from hypoxia? See, in hypoxia, we have a component of anaerobic glycolysis, which is not present in ischemia. As a result, ischemia is more rapid, leading to severe cell injury as compared to hypoxia. Coming to the next slide, let us look at the mechanism of ischemic cell injury. Any kind of reversible cell injury will lead to irreversible cell injury after 30 to 40 minutes of ischemia. I repeat, Any kind of reversible cell injury will lead to irreversible cell injury 30 to 40 minutes after ischemia, and it results in death mainly by process of necrosis. I repeat, it will result in death mainly by necrosis. So ischemic cell injury leads to death mainly by necrosis, but also there is a very small component of apoptosis. But for your single best answer, you will choose necrosis. If the options are necrosis, apoptosis, and third option is both of them, you will select both of them. Okay. Any kind of hypoxic stress leads to release of hypoxia inducible factor 1. I repeat, hypoxic stress leads to release of hypoxia inducible factor 1, which leads to three major changes. Number one, new blood vessel formation. Number two, anaerobic glycolysis. And number three, stimulates cell survival pathways. Repeat, hypoxic stress leading to hypoxia inducing factor 1 leads to new blood vessel formation, anaerobic glycolysis, and stimulation of cell survival pathways. Going to the next slide. Ischemia reperfusion injury is one of the very important mechanisms that we need to understand, mostly in case of myocardial infarction. Whenever blood supply to the injured part or to the ischemic part is restored, there is a paradoxical component of injury to that particular part. Let us understand how. When ischemia happens or ischemia occurs and we restore the blood flow, then two things can happen. Either there will be cell recovery if the cell is injured, or there can be paradoxical exacerbation of injury and cell death leading to what is called as reperfusion injury. Let me explain once again. Whenever there is ischemia to any organ, particularly in this case I am talking about the heart, and whenever the blood flow is restored to that particular organ, now there can be two events or there can be two results. Either this will lead to cell recovery if the cell is reversibly injured, or it can lead to paradoxical exacerbation of injury and cell death, ultimately leading to what is called as reperfusion injury. Let us see the mechanisms of ischemia reperfusion. There are four main mechanisms which we will be discussing in brief. Number one, oxidative stress. Number two, increased intracellular calcium. Number three, inflammation. And number four, stimulation of complement proteins. Let me repeat. There is oxidative stress, there is increased intracellular calcium, inflammation, and stimulation of complement proteins. Let us see number one, oxidative stress. So whenever a blood flow is restored to the ischemic part, it leads to an increased production of reactive oxygen species as well as reactive nitrogen species. Also, there is incomplete reduction of oxygen by the damaged mitochondria, and because of action of the oxidases which are present in the inflammatory cells in the region of ischemia. I repeat, there is oxidative stress because of increased amount of reactive oxygen species and reactive nitrogen species. Where is this reactive oxygen species coming from? It is coming by incomplete reduction of oxygen by damaged mitochondria, or because of action of the oxidases which are present in the inflammatory cells, which naturally is present at the site of ischemia. Second mechanism is increased intracellular calcium overload. As we have already discussed in the previous lectures, I will not be going into the details of this, but increased cytoplasmic calcium plays a role in ischemic reperfusion injury and leads to tissue damage. It begins during ischemia and it is more exacerbated during reperfusion. Third is inflammation. So how inflammation or why inflammation is causing ischemic reperfusion injury? Let us see. Now at the site of ischemia, some of the cells becomes dead and they will release certain danger signals. These danger signals are responsible for injury. Then there will be certain immune cells at that site which will release certain cytokines, which again plays a role in ischemia reperfusion, and any adhesion molecules released by the hypoxic parenchymal and endothelial cells. And as we all know, all these three signals, danger signals, cytokines, and adhesion molecules from the hypoxic parenchymal and endothelial cells, they will invite the neutrophils to come and create chaos at the site, leading to more injury at the site of ischemia reperfusion. Lastly, number 4 is stimulation of complement proteins. This is very interesting. Let us see. Now what happens at the site of ischemia? Antibody IgM goes and bind to the ischemic tissue. Now once that part is reperfused, the reperfused tissue bound to IgM, it stimulates the complement. Okay, and once the complement gets stimulated, it gets activated, leading to complement mediated injury to the ischemic part, which also accounts for ischemic reperfusion injury. With this, we complete ischemic reperfusion injury. Now going to third component, that is chemical injury. Now, chemical injury is very simple. They can be direct chemical injury, for example, by mercury as well as cyanide, or certain substances might get converted to toxic metabolites. For example, they can be free radical mediated injury to the cell, or for example, carbon tetrachloride can be converted to CCl3, which is toxic. Third example is acetaminophene getting converted to a toxic metabolite, which causes injury. All of these are examples for chemical injury. Now we start with a very important and interesting topic that is called as apoptosis. Each of these points are very important, and please pay careful attention as this is going to be very interesting. So apoptosis is defined as a programmed cell death. It is an irreversible phenomena. There are three very important things that we should know in apoptosis. One is, under apoptosis there is formation of something called as the apoptotic body, which is nothing but comprising of a portion of the cytoplasm and nucleus that is the fragments of the cell. So apoptotic body is nothing but containing fragments of the cell. This apoptotic cell finally will express certain receptors or certain molecules which will facilitate the phagocytosis of such cells. So apoptotic cell ultimately undergoes phagocytosis, and thirdly, because this process is so fast that there is no component of inflammation. So I repeat, there are three important things in apoptosis. Firstly, there is formation of apoptotic body, which is nothing but a portion of the cell constituents itself. Number two, there is apoptotic cell undergoing phagocytosis, and third, there is no inflammation. Now, why apoptosis occurs? It mainly occurs to remove the dying cells, aging cells, the harmful cells, the unwanted cells, and the diseased cells which are damaged beyond repair. I repeat, it occurs to remove the dying cells, the aging cells, harmful cells, unwanted cells, and diseased cells which are damaged beyond repair. Now, let us look at the examples of apoptosis. First, we will look at all the physiological examples in the first two slides, and then we come to the pathological examples. So under the physiological examples of apoptosis, number one are the cells that during embryogenesis, okay, they are continuously undergoing apoptosis. Okay, so this is physiological. Secondly, whenever there is loss of hormonal stimulation. So what are the examples? Involuting lactate of the lactating breast, right? So involution of lactating breast is one example. Ovarian follicular atresia following menopause is another example. Atrophy of the prostate after castration is another example. And endometrial loss at the end of our menstrual cycle is again one example. Going to the other slide for physiological example of apoptosis is the cell loss in proliferating cell population. Example, the epithelial cells in the intestinal crypts, they are continuously lost. Okay, so they are in proliferating cell population, the cell loss is always there, and that occurs by apoptosis. Elimination of potentially harmful self-reactive lymphocytes, also an example of physiological apoptosis. And lastly, the death of cells which has served its purpose. Okay, so these are the examples of physiological apoptosis. Coming to what is called as pathological apoptosis. Now, examples, whenever there is a DNA damage, maybe because of radiation or maybe because of anti-cancer drug, can lead to DNA damage and pathological apoptosis. Or whenever there is protein misfolding or unfolded protein response because of accumulation of certain misfolded proteins inside the cell. Again, an example of pathological apoptosis. Again, certain infections, viral infections like in adenovirus, HIV, all leads to apoptosis, as well as sometimes not the virus but the host immune response to the virus. For example, viral hepatitis can lead to pathological apoptosis. And lastly, pathological atrophy in parenchymal organs after duct obstruction. Okay, so pathological atrophy in parenchymal organs after duct obstruction. So here we go with the examples. Now, very important is the morphology of apoptosis. So listen very carefully. There is a cell shrinkage, and the most common characteristic of apoptosis is that there is chromatin condensation. The apoptotic bodies undergo phagocytosis, and there is cytoplasmic bleb formation. I repeat, there is cell shrinkage, chromatin condensation, apoptotic body formation ultimately leading to phagocytosis, and cytoplasmic bleb formation. Let us look at the histology of apoptosis. Now, these apoptotic cells are nothing but a round or oval mass of intensely eosinophilic cytoplasm. And apoptosis, as I have already discussed, is a very fast process, so it is not detected easily on histology. Okay, so before we come to the mechanism, let us look at certain diagrams to illustrate our points. Look at this very beautiful diagram. So this is a diagram of an apoptotic cell. Can you see there is a cell shrinkage, there is a nuclear or chromatin condensation, and look at the cytoplasm, which is intensely eosinophilic cytoplasm with a condensed nucleus, and there is a classical cell shrinkage. Look at this other diagram, which is a diagram of electron microscopy. Look how these nucleus are forming crescents at some places. Look how at some places they are condensed, and look how at some places they are undergoing fragmentation, which is also characteristics of apoptosis. Looking at other diagram, look at this diagram. This diagram is illustrating the formation of apoptotic bodies and also showing blebbing. This is a very good picture showing nuclear fragmentation, and this diagram illustrates the presence or activation of caspases, which leads to apoptosis. This is caspase 3 activation leading to apoptosis. Now, in short, I would like to give you a brief idea about the details or the mechanism of how apoptosis will occur or how the process takes place. Suppose there is any stimulus. So first of all, before I go into the details, there are two main pathways where there is a major pathway called as the mitochondrial or intrinsic pathway, and there is a minor pathway called as the death receptor or extrinsic pathway. I repeat, mitochondrial or extrinsic pathway, the major mechanism, and minor mechanism is the death receptor or extrinsic pathway. Now, look, any kind of stimulus, as we have read, maybe growth factor or decreased survival signals or DNA damage or protein misfolding, any kind of thing. They stimulate certain family of apoptotic proteins which are pro-apoptotic, means they cause apoptosis. These are nothing but the BCL2 family, that is back. Okay, and they will inhibit certain anti-apoptotic proteins, for example BCL2, BCLXL. Okay, this will ultimately lead to the release of cytochrome C and other pro-apoptotic proteins into the cytoplasm from the mitochondria. Ultimately, it will lead to initiation of caspases, executioner caspase leading to endonuclease activation, ultimately leading to nuclear fragmentation and breaking of the cytoskeleton. Both these processes results in the formation of cytoplasmic bleb, ultimately leading to apoptotic body formation, which I already said they comprise of nothing but portions of the fragmented cell, and ultimately these apoptotic bodies are expressing certain ligands which facilitates their phagocytosis. Okay, very simple process. Now, if you see in the mitochondrial process, okay, the death receptor pathway is also very similar, but this involves TNF receptor family as well as FAS receptors. Okay, and leads to the same initiator as well as executioner caspase and the same result, just the receptors are different. So here we go with the basics of mechanism, and now we will discuss them individually. So first, let us read about the mitochondrial intrinsic or the major mechanism. Before going into the details, we should discuss about the anti-apoptotic proteins, pro-apoptotic proteins, and the sensors and what are the roles of each. So number one, as we see, there are certain anti-apoptotic proteins, for example, BCL2, BCLXL and MCL1, which are present in the outer mitochondrial membrane. So there are anti-apoptotic proteins BCL2, BCLXL, MCL1, which are present in the outer mitochondrial membrane. Their major function is that they keep the mitochondrial outer membrane impermeable to the cytochrome, therefore it opposes apoptosis. Okay, so I repeat, as a name suggests, anti-apoptotic means they will oppose apoptotic, comprising of BCL2, BCLXL, MCL1 present in the outer mitochondrial membrane, keeping the mitochondrial outer membrane impermeable to cytochrome, therefore opposing apoptosis. Second group of protein that we know is the pro-apoptotic protein, which is the back and backs protein, having 4 BH domains, and this, as the name suggests, is pro-apoptotic. So they are forming channels in the mitochondrial outer membrane, and cytochrome C comes out stimulating the caspases. Okay, so they are forming certain channels in the mitochondrial outer membrane, letting out the cytochrome C and stimulating the caspases, ultimately leading to apoptosis. There are certain sensors. Okay, they are BIM, BID, BAD, PUMA, NOXA. Now their main function is that they are regulators of apoptosis, maintaining a balance between 1 and 2. So as and when it is required, it might shift towards pro-apoptotic, it might sometimes shift towards anti-apoptotic, okay, depending on what is required. Now coming to the main pathway, the major mechanism, that is the mitochondrial intrinsic pathway. As we have already seen, it starts with the cytochrome C in the cytosol coming from the mitochondria. This cytochrome C binds to apoptosis activating factor, also called as APAF, and this APAF forms what is known as apoptosome. Now this apoptosome finally binds to caspase 9 and it will undergo cleavage to form active caspase 9. It will undergo cleavage to form active caspase 9, and this is the initiator caspase. This is the initiator caspase. Again, I repeat, the apoptosome binds to initiator caspase 9, leading to its cleavage, giving rise to active caspase number 9. Now this will trigger a cascade of caspase activation by cleavage of pro-caspases. Okay, so this initiator caspase, that is 9, is now going to stimulate other pro-caspases, ultimately leading to apoptosis by the executioner caspase. Now, little thing that I would like to mention over here, that in our body there is something called as SMAC and diablo, which are just like cytochrome C, and once they come into the cytosol, they also inhibit the physiological inhibitors of apoptosis, ultimately leading to apoptosis. This is just an extra information that I am giving you. Till here you should understand nicely. Okay, now this is what I have already explained you, leading to the cytochrome C coming out of here, leakage is there, activation of caspase leading to apoptosis. Now we come to the second mechanism, that is the minor mechanism of apoptosis, that is the death receptor initiated or the extrinsic pathway. Okay, so as we know, the death receptor is a member of the receptor TNF receptor family. The death receptor is having two domains. Okay, one is the cytoplasmic domain and another is the outer domain. I will show you in the diagram. Look over here. There is a cytoplasmic domain, that is the death domain, and there is an outer domain. Okay, an example of the receptors, as we see on the cytoplasmic domain, is the type 1 TNF receptor as well as Fas associated death domain. I repeat, the examples of cytoplasmic domain, that is also called as the death domain, are type 1 TNF receptor and Fas associated death domain. Now the outer domain, the receptor, the example of that is Fas, that is CD95. Let us look over here. The Fas is an example of outer domain, and the death domain, that is FAT, is an example of cytoplasmic domain. Okay. What is important over here that once the FAD will get stimulated, it will act on pro-caspase 8 leading to formation of active caspase 8 leading to stimulation of executioner caspase ultimately leading to apoptosis. I repeat, FAD will lead to stimulation of pro-caspase 8 leading to of caspase 8. Ultimately, they will stimulate the executioner caspase bringing about apoptosis. Now, there are two important points that I would like to share at this point, that the FAST receptor is expressed in the self-reactive T lymphocytes. So self-reactive T lymphocytes, as we have already read, is an example of physiological apoptosis. And by which pathway they will occur apoptosis? By the death receptor initiated or the extrinsic pathway they will undergo apoptosis. Inhibitor of apoptosis, that is the intrinsic pathway, there is some protein which inhibits, those are called as the flip proteins. If you look at this pathway, as I have already discussed, the FAD leads to activation of pro-caspase into active caspase, leading to stimulation of executioner caspase, ultimately leading to apoptosis. Now, finally, let us look at the execution phase of the apoptosis. So, as we see, mitochondrial or intrinsic pathway, the initiator is caspase 9, and death receptor or extrinsic pathway, the initiator is caspase 8 and 10. I repeat, the mitochondrial or initiator intrinsic mechanism, the initiator caspase is 9, whereas the death receptor or extrinsic pathway, the initiator caspase is 8 and 10. Finally, the execution phase by the caspase is brought about by a common caspase, that is caspase 3 and 6. So execution phase of the caspase is brought about by the caspase 3 and 6, ultimately leading to endonuclease stimulation, as I have already said, and breakdown of cytoskeleton. Both of the process leads to DNA cleavage and nuclear fragmentation, ultimately leading to cytoplasmic bleb formation, as I have already explained, leading to apoptotic body formation, which undergoes phagocytosis. So cytoplasmic bleb formation, apoptotic body formation, and phagocytosis. And as I have already said, since the process is very fast, there is no inflammation and there is no necrosis that can be discernible. With this, we end the apoptosis. Coming to certain disorders which are associated with dysregulated apoptosis. Let us see. So increased apoptosis is seen in certain neurodegenerative disorder as a part of ischemic injury and also death of virus infected cells. Okay, I repeat, increased apoptosis is seen in neurodegenerative disorder, ischemic injury, but we have to keep in mind the predominant form is necrosis and also some parts of ischemic injury is apoptosis. Death of virus infected cells. Now, decreased apoptosis. Let us look at decreased apoptosis. The most common genetic abnormality is P53 mutation. There is no apoptosis, and there is accumulation of different kind of mutations, and once these abnormal mutations accumulate, that will lead to cancer. Okay, failure to eliminate the self-reactive lymphocytes leading to autoimmune disease. There are two main things. So most common genetic abnormality of decreased apoptosis is when there is P53 mutation. So if there is no death by apoptosis, the abnormal cells or damaged cells will harbor the defective DNA, mutations will accumulate, ultimately leading to what? Cancers. This is one example. Another example is failure to eliminate self-reactive lymphocytes, which leads to autoimmune disease. Now there are two very small topics, interesting topics, one of which is necroptosis. Now necroptosis is also known as programmed necrosis. Okay, it has features of both necrosis as well as apoptosis. Morphologically and biochemically, they are just like necrosis, but mechanistically like apoptosis. I repeat, morphologically and biochemically, they are like necrosis, whereas mechanistically, it is like apoptosis. How is it different from apoptosis? Because there is no caspase activation. Therefore, it is called as caspase independent cell death. But it begins, the death pattern begins as extrinsic form of apoptosis, that is the death receptor pathway via it starts. But cell death ultimately occurs like necrosis. For example, as we have seen in the beginning of our lecture, reduced ATP and increased ROS production. Now important for necroptosis are two proteins, one is RIP1 and RIP3. let us look at certain examples of necroptosis. Number 1 is steatohepatitis. Number 2 is acute pancreatitis. Number 3 is reperfusion injury. Number 4 is neurodegenerative injury. Number 5 is against viral infection. So examples of necroptosis are steatohepatitis, acute pancreatitis, reperfusion injury, neurodegenerative injury, against viral infection. And last topic for the day is pyroptosis. As the name suggests, it is defined as cell death which is associated with fever-inducing cytokine interleukin 1. So it is associated with fever-inducing cytokine interleukin 1. So microsomal products and innate immune receptors, both of them ultimately leads to inflammasome formation, which stimulates caspase-1, interleukin-1 beta convertase enzyme. This cleaves the precursor interleukin-1 and activates interleukin-1, leading to cell swelling, loss of plasma membrane integrity, and release of inflammatory mediators. So with this, we end this lecture. Hope you like my video. Please subscribe to my channel. Please like the videos if it helps you, and please spread the message. Thank you so much. Here is Jibran Ahmed with you, and stay tuned for further videos. Thank you so much.